Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is transporting plant, and in that we have already discussed about the short distance transport, and in long distance transport also we have talked about the symplast, apoplast, then root pressure and transpiration pull theory. And when we have already studied about the transpiration pull theory, we have know that transpiration, that is a loss of water in the form of vapor. But we have to know something more about the transpiration that we will discuss in this today's lecture. So transpiration, you have studied transpiration in earlier class by enclosing a healthy plant in a polythene bag and observing a droplet of water from inside the bag. Okay, you can see the when you will take a healthy plant and cover it with a polythene bag, you will see the observe the droplet of water inside the bag. So this droplet of water are ultimately indicating that the parts of the plant releasing water. So you could also study water loss from leaf using a cobalt chloride paper which changes color on absorbing water. So this is the one cobalt chloride test which also help us to determine whether transpiration is occurring or not. So it is a simple experiment that we carry out here. A leaf is taken, okay, and on the leaf the cobalt chloride paper on upper surface is kept, and with the rubber band it is tied in a proper way on the glass side, on the both sides have kept, and then it is tied with the rubber band. So this cobalt chloride paper cannot shift, okay, and it is a leaf from the potted plant. Now cobalt chloride paper on the lower surface as well, that is a bluing color, on the upper surface also it is a bluing color, and both side the cobalt chloride paper is also covered by the rubber cobalt chloride also glass side, and then tied with the rubber band. Now, when the transpiration will occur, a water loss will occur, and the paper turned less pink, or paper turned more pink. Okay, on the back side, the paper will turn more pink, and on the upper side, you will see the paper is turned the less pink. So, this conversion from blue to pink is indicating that the cobalt chloride is coming in contact with water, and due to which it changed into the pink color. So, it is showing that the water is lost from the particular leaf surface. So from leaf using bell jar experiment, also we can understand this, where you can see drops of colorless liquids inside the bell jar containing the plant. You can see this is the control plant, it is a potted plant, and here you can see it is a control pot is there. Now we have kept a plant. Now after covering the plant, we are covering it with the bell jar. You will see when we put a plant, you will see that water drops on the surface of the bell jar. So this also indicating that the water is losing from the plant surface. So this water loss, if occur in the form of vapor from the living tissues of the aerial parts of the plant, then it is known as transpiration. So what is transpiration? It is a loss of water in the form of vapor from the living tissues of the aerial parts of the plant. And what was guttation which we have discussed? There is a loss of water in the form of liquid droplets. So this is a transpiration. Okay, and how the water loss is occurring in the form of vapor. And it is known that this transpiration play a significant role in concept of SPAC, that is the soil plant atmosphere continuum. Means the water is absorbed from the soil, it goes within the plant, it move up in the plant, and from the aerial surface it is losing to the atmosphere. So this is the continuum from atmosphere, again through rain it will reach to the water, to the soil, and then it will come again within the plant, and the cycle goes on continuing. So what is the transpiration that we have discussed already? Loss of water in the form of vapor from the living tissues of the aerial parts of the plant. Now the water absorbed by the plant from the soil through the root system is lost into the atmosphere through the shoot system. So absorption is done by the root system, but loss occurs through the shoot system, which in turn gets back to the soil through the base. Okay, so this is the continuum, how the particular spec that the soil at plant atmosphere continuum take place. So through rain Means you can see the water will reach to this water body. From here it will reach to the plant. From plant it will reach to here. From here it will reach to atmosphere, and that water will again come through this to the soil. So the phenomena of transpiration occurs mostly through the stomata. Mostly, the water loss in the form of vapor occurs through the stomata, and the term stomata is a plural. Singular term is stoma, and this stoma are located on the leaves in the epidermis. And through that stoma on the epidermis, the water loss occurs mostly in the form of vapor. So you can see this is stomata. Those opening and closing of stomata take place. When stomata get opened, the water loss occur, and when stomata get closed, water loss does not occur. Okay. So water loss occur in the opening stage. So now, first we have to discuss the types of transpiration. When we talk about types of transpiration, the first type is stomatal transpiration. And if we talk about a stomatal transpiration, it is a transpiration that occur through stomata. So here, transpiration occur through stomata. Okay, so it is called as stomatal transpiration. And it is also important that stomata are mainly present on leaves, mainly present on leaves, and also also present on delicate organs like sepals, herbaceous plants, herbaceous stems specifically. Here we are going to talk about what organ. So we write here herbaceous stem. Okay, so this through this the particular this take place, and it is 50 to 90% of total transpiration. Means whatever the total transpiration, out of this 50 to 90% of the total transpiration it is accounted by this stomatal transpiration or occurs through this stomatal transpiration. Okay, so that is the first one. Now after this, the second type of transpiration is known as cuticular transpiration. And this cuticular transpiration, if you take, there is a loss of vapor via thin cuticle. Loss of water vapor via thin cuticle, then it is known as cuticular transpiration. We know that cuticle is a waxy layer present on the epidermis of the aerial parts. Okay, so through this thin cuticle, if the loss of water is occurring in the form of vapor, then it is a cuticular transpiration, and it accounts for 9 to 99%, 9.9% of the total transpiration. Total transpiration. The data may vary. So it at some places it is written as 1 to 10%. Okay, so that much also it can be given in this particular one. Okay, so and whatever the stomatal transpiration we have written, that is later given us as a 50 to 90%. Obviously, that can be 97%. This critical transpiration to the critical, it is 9 to 9 or 3 to 10% of total. But in herbs and in herbs and ferns, it may be 50%. Okay, so such account can be there. Third is bark transpiration. This bark transpiration, if you talk, that occur through bark. So whatever the spaces left in bark, if through that if transpiration occurs, then it is known as bark transpiration, and it is approximately 1% of total transpiration. Apart from this, the fourth one is lenticular transpiration. And we know that lenticel are the breathing pores through which the transpiration takes place. So this lenticular transpiration, it is a loss of water vapor from lenticels or aerating pores in bark of tree or fruits, etc. And it is hardly 0.1% of total transpiration. Okay, so these are the all the particular types of transpiration based on the structure which is involved in transpiration, like the particularly stomata, bark, lenticel, and this thing. Now, now after this, one more term is given, that is foliar transpiration. So here the foliar means the leaves. Okay, so it is the total transpiration. Foliar transpiration is the total transpiration takes place through surface of leaves, that is stomatal and cuticular. Through this, the whatever the transpiration occurring through the leaf, because cuticle and stomata are the result of the leaves. So if through that the transpiration occurs, then it is known as a what? A particularly foliar transpiration. Okay, so this is the all different types of transpiration. So the majority transpiration occurs through which one? A stomata. So if we talk about stomata, stomata in most plant are photoactive stomata. In majority plant, you will see the presence of photoactive stomata. Now, photoactive stomata are those stomata which are open during the day and closed during the night. So this is the type of stomata. Okay, so you can see this is the stomata and this is the open stomata, whereas this is the closed stomata. Okay. Now, There are one more type of stomata which is known as photoactive stomata. So in a succulent plant, or we can say in camp plant, succulent plant means water holding xerophytic plants. It is noted that the transpiration occur at night through this photoactive stomata, because this photoactive stomata are open during night and remain closed during the day. They are doing a reverse mechanism. So this photoactive stomata are found in some succulent xerophyte like cactus, bryophyllum, opuntia. Okay, we can say right a pineapple. These all particular different examples. We can say they have this protective stomata which are open during night and closed during the day. So besides transpiration, exchange of oxygen and carbon dioxide also occur through the stomata. Stomata is not playing only role in the transpiration. It is also carrying out the exchange of carbon dioxide and the oxygen through it. So this is the stomata where the CO2 intake takes place and oxygen outgo occur. So CO2 getting absorbed by the stomata and O2 is released. So this exchange takes place through stomata also. So usually the lower surface of the dorsiventral leaf, dorsiventral means they are often dicotyledonous leaf. They have greater number of stomata on the lower surface. While in isobilateral leaf, there are the monocot leaf, they have nearly equal stomata on both the surface. That also we have studied. So transpiration is going to be affected by several factors. Several external factors affect this. So that is the temperature. Okay, then we can say light, humidity, wind speed, etc. They may affect this particular transpiration. So if temperature is affecting the transpiration, up to a certain limit, it increase in temperature results in increase in rate of transpiration. At a very high temperature, transpiration decreases because it's going to be affect or kill or damage the particular transpiring tissues or the organs. So these factors which affecting the transpiration, these all are the external factors. Okay, now how they are affecting that we have to discuss that. External factor in this, the first is temperature. So we have shown that as the temperature increases, the rate of transpiration increases, but up to a certain limit, not beyond that. So this is directly proportional. Increase in temperature is directly proportional to the rate of transpiration, because if the temperature is getting increasing, then the vaporization will occur, and obviously the loss will occur in the form of what? A transpiration. Next is the light. If you talk about a light, it also stimulates transpiration. So it also stimulates transpiration by favoring opening of stomata. Okay, through this light particularly affect. And in that, the blue light is slightly more effective than the red light. Okay, in case of the particularly this transpiration. Next is the humidity, that is atmospheric humidity. If you talk about atmospheric humidity, it is the most important factor. If the atmospheric humidity is high already in the atmosphere, then obviously transpiration is not going to occur. So humidity is inversely proportional to the rate of transpiration. If humidity is high, the transpiration will decrease. Okay. And apart from this, the next factor is the wind speed or wind velocity. So transpiration is less in still air. Actually, it is considered less in still air, and the rate of transpiration increases with the increase in wind velocity. Okay. So wind velocity, in that also there is a two things. One is slow wind. We call it a slow breeze, and the slow breeze is considered as 5 to 50 kilometer per hour, and this increases the rate of transpiration. Now why it is increasing the rate of transpiration? Because suppose the leaf surface is there, okay, and over leaf surface some salts have been accumulated, and due to this stomata are not carrying out a transpiration, so at a slow breeze this all will be removed. Okay, and it will be removed due to this transpiration will increase. But if the wind speed become too much high, very high wind velocity, At that time, the point will come that this leaf will fall down. So at very high velocity, high wind velocity, rate of transpiration decreases. Rate of transpiration decreases. Okay, so this is the factors. Now that is external factor. Now how the internal factors or which internal factors affect the, particularly we can say the rate of transpiration or a transpiration. So plant factor, or we can say internal factors that affect the transpiration include, first is the number and distribution of stomata. So number of stomata is obviously increasing. Then if the more number of stomata are there, then the rate of transpiration will increase obviously. Okay. And if we talk about the distribution, if a distribution of stomata is variable, usually on the lower surface in dicot stomata, the number of stomata are greater, as you have discussed. And in isobilateral, both surfaces have same. So transpiration, monocot transpire more than the dicot. Okay. So in this, number of stomata is directly proportional to the transpiration. It increases, the transpiration increases. But according to the distribution of stomata in the monocot and dicot leaf, it is also known that monocot transpire more than dicot. Then next we talk about the percent of open stomata. Obviously, if the number of open stomata are more, the transpiration will be more. So it is directly proportional to the rate of transpiration. Then water status in the plant is obviously the water is more in the plant than the transpiration will occur. And we know that less than 1% of the water reaching the leaves in the use is used in photosynthesis and plant growth. And most of absorbed water lost through the transpiration. Thus, a good status of water favor the rate of transpiration. So, water status of the plant. So, good status of water favor transpiration rate. Okay. Next, canopy structure. If we talk about a canopy structure, the dense canopy causes a decrease in transpiration because the canopy obstruct the airflow. So if a dense canopy is there, okay, then we remember dense canopy decreases the rate of transpiration because canopy obstructs the airflow. Then fifth is available soil water. Obviously, if the soil water is more than the available soil water is more, then it is directly proportional to the rate of transpiration. Because if the available soil water is more, the absorption will be more, absorption will be more, then the loss will be more. And the sixth is the root shoot ratio. If we talk about root shoot ratio, this is directly proportional to the per unit area. If we describe, it is directly proportional to the transpiration rate. And in root shoot ratio, the dry weight of root and dry weight of shoot is taken, and the ratio is taken. If this ratio is increasing, then the transpiration rate is increasing. Okay, so this is the how the particularly we can say this effect. So after the internal external factor which affecting the plant or transpiration, now we are going to talk about the mainly that transpiration is occurring through the stomata. Then we have to understand that how this stomata opening and closing take place. So immediate cause of opening or closing of stomata is the change in the turgidity of the guard cell, and turgidity explains the water status of the plant's guard cell. So if the water status is less in the guard cell, the means the water is released, and due to this the water status will be less, then the stomata get closed. If in this guard cell water is getting absorbed, then water status will be more, and if the water status is more, the guard cell will swell, and due to this the stomata will open. In guard cell, if you take inner wall of each guard cell towards the pore or stomatal aperture are thick and elastic. If you will see wall, this wall are thick and elastic, whereas this wall, these are thin and inelastic as NCRT. But it should be elastic, should be the outer one. Okay. So here in this particularly thing is observed in the guard cell. Now, what happens when the turgidity increases, means the water content increases within the two guard cells, the flanking each stomatal pore or pore, the thin outer wall bulge out. And when the thin outer wall bulge out, the force the inner walls into the crescent shape. This will force the inner cell to convert into this crescent shape, and due to this, the opening of stomata will occur. So first, the water status will be increased. Water will increase in guard cell. The water will push the outer wall to come to bulge out, and when the outer wall bulge out, it will take with it the inner wall and form a crescent shape of this inner wall. So the opening of stoma is also aided by the orientation of the microfibril in the cell wall of the guard cell. So cellulose microfibrils are also found in the guard cells, and they are oriented radially rather than longitudinally, making it easier for the stoma to open. So this is the actual structure. You can see this is the radially arranged like this, radially oriented cellulose microfibril, and they are somewhat rigid. Because of this, when outer wall goes bulging out, the inner wall also along with the outer wall go in a crescent shape. They have this cell wall. The inner wall is thick, inner outer wall is thin. You can see these guard cells are there. These guard cells have, if we talk, they have the chloroplast. Okay, they have vacuole. Okay, these all things are found, and surrounding to this guard cell you will see the presence of the subsidiary cells, and this is the stomatal pore, or we can say stomatal aperture. So this way the things are found, okay, in the stomatal structure, and this is specifically a dicot stomata. So when the guard cells lose turgor due to water loss or water stress, the elastic inner wall regain their original shape, and the guard cells become flaccid and the stomata close. So when water loss will occur, the guard cell become flaccid and the stomata closes. So you can see this is the water intake is occurring, and due to water intake, the guard cell are getting swollen and or turgid, and the stomata is getting open. Okay. Now when the water loss occur, the guard cell will again come to their original shape, and the closing of stomata will take place, and that is shown in this diagram clearly. So now this opening and closing of stomata, as we have explained with the simple theory, we have to understand that in the proper way, and that we have to understand the mechanism of opening and closing of stomata, and this mechanism of opening and closing of stomata is specially explained by the Levitt, and who proposed one important theory, that is a potassium pump theory, to explain the mechanism of opening and closing of photoactive stomata. So according to this theory, accumulation of K plus ions takes place into the guard cell from the subsidiary cells, and this occur in the presence of light. Okay, so this coupled with the efflux of proton leads to the increase in pH of the guard cell. And now how this occur, that we should understand by here with this diagram. So if we talk about the potassium pump theory, dear student, suppose let's draw one guard cell. This is guard cell. Now what happens first in guard cell? You will see that there is a carbohydrate or starch is there in the guard cell. Now this will convert into first glucose. Then this glucose undergo EMP pathway or glycolysis and converted into PEP, that is phosphoenol pyruvate. And this phosphoenol pyruvate is a 3 carbon compound. So this 3 carbon compound accepts CO2 from the atmosphere. As it accepts CO2 from the atmosphere or the surrounding cells, it converted into oxaloacetic acid. Now this oxaloacetic acid is of 4 carbon, and this oxaloacetic acid further converted into malic acid. This malic acid is also of 4 carbon. Now this malic acid is broken up into malate and H plus. Now this H plus goes out, and along with same antiporter, the K plus will come inside. Now this K plus react with this malate, and when this K plus react with this malate, it is going to form a potassium malate. Now this potassium malate enter the vacuole, and in vacuole, as the potassium malate increases, the solute concentration is getting increased. So osmotic pressure increases, and osmotic pressure increases, due to which endoosmosis will take place and water will come inside. And due to this, the guard cell become turgid. And when guard cell become turgid, the stomata open. Okay, so how this the particularly solute concentration of potassium will get increased, and due to this the endoosmosis will take place and the water content will increase in the guard cell, and ultimately guard cell will become turgid, and due to this the opening of stomata will occur. Okay, so how this the particular opening of stomata takes place in the potassium pump theory, which is explained by the Levitt. Now, if some more K plus comes inside to equilibrate that, the chlorine will also come inside extra, and that chlorine will react with the potassium and from KCl, due to which the potassium will not increase in too much amount. Now, this is the opening. Now, after opening, the closing of stomata also occur. And in closing of stomata, the modern theory explained that is known as abscisic acid theory. And according to abscisic acid theory, it is known that ABA concentration increases, sorry, CO2 concentration. CO2 concentration increases in substomatal cavity. Substomatal cavity is the cavity. If suppose this is the leaf, here stomata is there, here some mesophyll cells are there. This cavity is known as substomatal cavity. So the cavity which is present behind to the stomata, it is known that substomatal cavity. Now, when the CO2 concentration increases in the substomatal cavity, what it means? Abscisic acid synthesis occur in guard cell. Now, this abscisic acid changes the permeability of guard cell for K plus and cause efflux of K plus from guard cells. When K plus potassium malate will break down and K plus will go out from the guard cell, so the osmotic pressure for solute get decreased and osmotic pressure getting decreased. So osmotic pressure will decrease, so exoosmosis will occur and the stomata get closed. So how this the particularly interfere with the K plus A plus H plus pump and carry out a closing of stomata, that is abscisic acid, which is a modern theory. Okay, so how the opening and closing of stomata occurs. So accumulation of K plus we have discussed in the guard cell associated with the passive influx of a chlorine ion, thereby decreasing the water potential of the guard cell. Water thereby enters the guard cell, making them turgid and the opening occur. The outer walls are thin and elastic in the guard cell that we have discussed. Guard cell expand outwardly, leaving a minute pore in the center open, that is a what? A stomatal pore. So K plus will come inside and the opening will occur. At night, in the absence of light, K plus and chlorine ions move out of the guard cell, due to which the water potential of the guard cell increases and water start moving out of the cell, which leads to the closure of stomata. That also we have discussed. Under water stress condition, means water is less, the abscisic acid and anti natural anti-transparent drives the K plus ions out of the guard cell, making them close. In succulent plant, the water potential gradient establishes due to the accumulation of organic acid at night, make the guard cell become turgid. Okay, so in succulent plant, they open because stomata open during night because they have stochastic stomata. Why? Because the organic acids are accumulated at night in the guard cell, hence become turgid and stomata become open at night. Okay, so this is the thing about the what, particularly we can say the transpiration opening and closing of stomata. Now we have to discuss some more point with respect to transpiration, and that are the some facts about transpiration. So if we talk about some facts about transpiration, then the first thing we have to talk about the anti-transpirant. So first the question is, what are these anti-transpirants? So these are the substances which reduces the rate of transpiration, which reduces the rate of transpiration, which are known as anti-transparent. And if we talk examples of this anti-transparent, they include phenyl mercuric acetate, and this phenyl mercuric acetate is a PMA. Then the another one is the aspirin, that is salicylic acid. Then EBA, that is abscisic acid, a plant hormone. Then silicon oil, CO2, and low viscosity. It is not it is viscosity. Waxes. Okay, so that are the particularly we can say the anti-transpirants, which are going to reduce the transpiration. Now next we are going to talk about the one more term which is known as transpiration ratio. If you talk about transpiration ratio, it is a rate of the loss of water to the photosynthetic CO2 fixed is called transpiration ratio. So transpiration ratio is equal to moles of H2O transpired upon moles of CO2 assimilated or fixed in the synthesis of glucose. That is the transpiration ratio. Okay, so it is a, because stomata is playing a role in the transpiration as well as the CO2 absorption. So the how whatever the mole amount water transpired with respect to how much photosynthesis occurred, that is the transpiration ratio. So this ratio is low in C4 plants. So C4 plants are the temperature tolerant plant. We will discuss in photosynthesis chapter. They have 200 to 350 value, while C3 plants have 500 to 1000 value of a transpiration ratio. So this indicates C4 plant have a high low transpiration ratio compared to the C3, means indicate that C4 plant plant conserve water with efficient photosynthesis. And we will discuss in photosynthesis that the ratio of one type of plant are that chem plant, which has a minimum transpiration that you ratio that is 50 to 100. Okay, so this is the thing which is seen in case of, we can say, the this transpiration ratio. In xerophytes, obviously they have sunken stomata and they minimize the loss of water. Apart from this, two more statements are given by different scientists. And that is, it is known that according to Curtis, transpiration is an essential evil. Transpiration is an essential evil. So transpiration is an essential evil means in transpiration the water loss will occur at a maximum rate, we can say. Okay, in transpiration loss occur obviously, but if transpiration will take place, then the water will be given for the, we can say, the photosynthesis. Because of this we call it as the essential evil. And according to Stewart, transpiration is unavoidable evil. Okay, because at higher humidity, although transpiration is negligible, but the stomata remain completely open. And if the stomata are open, the transpiration is going to occur if when the temperature increases. So if temperature increases, the transpiration is going to be, so it cannot be avoided. Okay, so because of this, it is known as unavoidable evil, and that is what we can say some facts. So after the types of transpiration and the transpiration and opening and closing of stomata, now we are going to discuss that if transpiration is occurring, the water is to be applied for the photosynthesis, and photosynthesis water is necessary. So how we consider this photosynthesis and transpiration as a compromise? So it is a one NCRT topic. So we know that transpiration has a more than one purpose. It creates transpiration pool because transpiration pool will develop due to transpiration in the leaf, and due to transpiration pool, the absorption and transportation in plant take place. Then due to this transpiration pool, it supplies the water for the photosynthesis, transports minerals from soil to all parts of the plant. It cools leaf surfaces. Okay, the cooling effect is one of the important effect. It cools leaf surfaces, sometimes minimize. 10 to 15 degrees Celsius by evaporative cooling. So this way the cooling also takes place in this. This transpiration also maintains the shape and structure of the plant by keeping cells turgid by making available the water. So when we talk about the actively photosynthesizing plant has an insatiable need of water because they need a water continuously for their photosynthesis. So photosynthesis is limited by availability of water, which can be swiftly depleted by transpiration. So if photosynthesis need to occur, it can be only limited by the availability of water, and availability of water can be water will be limited or deplete if the transpiration will not occur. So to photosynthesis to occur, transpiration has to occur. So the humidity of the rainforest is large due to the vast cycling of water from the root to the leaves to the atmosphere and back to the soil. And despite making plant lose substantial amount of water, transpiration is beneficial plants in many ways. So first we are talking about this particular. So an actively photosynthetic plant has insatiable needs. So photosynthesis is limited by availability of water. Okay, and that we have discussed. Now in this one more thing happened, that as the evolution occurred, it is known that the evolution of the C4 photosynthetic system is probably one of the strategy for maximizing the availability of CO2. The evolution of C4 photosynthetic system is probably the one of the strategy for maximizing the availability of CO2 while minimizing the water loss because they are tolerant to the high temperature. So C4 plant are twice efficient as C3 plant in terms of fixing carbon because in C3 plant we will discuss that the carboxylation cannot occur always because the Rubisco enzyme will act and Rubisco can act as carboxylase and oxygenase. But since C4 plant, the first CO2 fixation will take place by the peptides which carry out only carboxylation. So because of this, C4 plants are twice efficient than C3 plant in fixing the carbon. This is not can be easily understood here, but it will be more understood in the photosynthesis chapter. So C4 plant loses only half much water as compared to the C3 plant for the same amount of CO2 fixed. Because of this, C4 are beneficial than the C3. Okay. Now, dear student, we remember two instruments are there that you should remember. One is photometer, and it is known that photometer is apparatus. Photometer is an apparatus used to measure rate of transpiration. And this apparatus works on the rate of water absorption is equal to the rate of transpiration. So photometer instrument which measures the rate of transpiration. And one more is there which is known as porometer. It is known that porometer, an apparatus used to measure area of stomata only, used to measure pore size of stomata on leaf. Okay, so this is the what we can say the transpiration. So exchange of CO2 and O2 takes place through, obviously it is occurring through the stomata along with the transpiration. Photoactive stomata open during daytime and closed during night. K plus pump theory was proposed by obviously Levitt that we have discussed. Okay, so this is the what we can say the transpiration and related thing. Now, in this uptake and transport of mineral nutrients, if we talk that we will discuss in mineral nutrition. But be dear student, uptake and transport of mineral nutrient, if we talk, plants obtain their carbon and most of their oxygen from CO2, obviously, in the atmosphere. However, the remaining nutritional requirement are obtained from the minerals and water in the soil. So majority minerals are obtained from the soil. Okay. So this atmosphere, O2 and CO2 are provided this, but soil will provide other all elements. So uptake of mineral ions, if we take, unlike water, or minerals cannot be passively absorbed by the root, because water will be absorbed by the diffusion or osmosis, but minerals are not going to be absorbed passively. All minerals, yes, majority minerals are absorbed actively, but not all minerals are absorbed passively. So two factors account for this: that majority minerals are absorbed actively, that minerals are present in the soil as a charged particles which cannot move across the membrane. And second, concentration of minerals in the soil is usually lower than the concentration of minerals in the root. So if the concentration of minerals in the root is high, the water will move from the soil to the root, the mineral will move from the soil to the root, then it is going to be against concentration gradient. So there is a need of active transport. So cation retention on soil clays will occur. So therefore, most minerals, because the particularly are the charged particles, therefore most minerals must enter the root by the active absorption into the cytoplasm of the epidermal cell, and this needs energy in the form of ATP. Obviously, majority minerals are absorbed by the active transport. The activity of ions is partly dependent responsible for the water potential gradient in root, because when the minerals are absorbed in the root, then the concentration is increases, then the water potential is going to be decreased because solute is increasing. Therefore, the uptake of water will take place by the osmosis. So some ions also move into epidermal cells passively, but majority move by the active transport. Ions are absorbed from the soil by the both passive and active transport. Specific proteins in the membranes of the root hair cell actively pump ions from the soil into the cytoplasm of the epidermal cells. So special proteins are involved, and they are involved in active transport, and ATP use also occurs. Like all cells, the endodermal cells may have transport proteins. Many transport proteins are embedded in their plasma membrane, and they lead some solute to cross the membrane, but not others. So it is acting selectively, the endodermal membrane. So the transport proteins which are present in endodermal cells, they are considered as a control points. Important point. This you should remember. They will ask majority time the question from this, where plant is going to adjust the quantity of the solutes and the type of solute that are going to reach the xylem. Note that the root endodermis, because of the layers of suberin, has the ability to actively transport ions in one direction only. Okay, the root endodermis, we know that have a Casparian strips, and Casparian strips have a suberin deposition, and that will allow the transport of ions actively in one direction only. That is from the surface or outside the soil to the epidermis, and from epidermis to cortex, cortex to endodermis, endodermis to so towards the xylem they will allow, but from xylem they will not allow the direction of movement of these ions. Okay, now translocation of mineral ions, after that ions that have reached the xylem through the active or passive or a combination of the two, their further transport up the stem to all parts of the plant is through the transpiration stream. So along with the transpiration stream, along with the water, they are going to move upward in the plant. Now the cheap sink for the mineral element, where the mineral element is required mainly, they are the growing regions of the plant, such as apical and lateral meristem, young leaves, developing flowers, fruits and seeds, and the storage organs. These are the young elements which are acting as a cheap sink. Unloading of mineral ions occur at the fine winding endings through the diffusion and active uptake by the cells. When the ions are unloaded, they will unload it from the vein endings by the diffusion, but the cell will take by the active transport. So the minerals ion are frequently remobilized, particularly from older senescing parts. Okay, and older dying leaves export much of their minerals content to the younger leaf. Obviously, the older dying have no role of this mineral, so they will export to the younger tissues. Before leaf fall in deciduous plant, minerals are removed to other parts, obviously, and elements most readily mobilized are phosphorus, nitrogen, potassium, and Pk. Okay. Some elements that are structural component like calcium are not remobilized. They are not going to be remobilized easily. An analysis of xylem exudate shows that though some of the nitrogen travel as inorganic ions, majority of the nitrogen or much of the nitrogen is carried as an inorganic form as amino acid and related compounds through the xylem. So similarly, small amount of phosphorus and sulfur are also carried as organic compound. Small amount of exchange of material takes place between the xylem and phloem. So hence we cannot clearly make that much of the organic nitrogen is going through the xylem. So we cannot make a clear distinction that xylem transport only inorganic nutrients and phloem transport only organic materials. Okay, so that is the water particularly thing. So epidermis cells needs energy in the form of ATP of this. Ions are absorbed from soil by active and passive both, but mainly active. Phloem transport, both inorganic and organic, and that is all about the water, particularly mineral absorption and translocation. Okay, I hope you understood that. Thank you.